Hello, welcome to another OmniFocus Fitness video. Welcome to another OmniFocus Fitness video. Today we will be discussing glycolysis. So another metabolism video, and you've probably noticed how long this video is. Well, have no fear, I will be taking care of that in this video. Yes, this video is extremely detailed and it does go over glycolysis, which you're probably thinking, well, why wouldn't I just watch a 15-minute glycolysis video that I can just find on another channel? Well, there's a good reason for that. Mainly because not only do I go into far more detail in this video than you probably will find in any other video, but I explain why glycolysis occurs. And I think that is so important for us to understand, why does glycolysis occur? Why does glucose have to go through 10 different steps for it to come out with a particular product? Why? And why is each step set up the way that it is? Well, I will detail why in this video for each single step, so you will understand, you will have a full, comprehensive view of each biochemical step, as well as further information related to glycolysis, how it occurs, where it occurs, why it occurs, why we care, what happens after glycolysis, all covered in this video, all scientifically backed in the article that is linked below. If you'd like to see more details, which is hard to imagine because this video is really long, there are more details in the article linked below, all free information. Now, of course, this video is quite long, as I've already mentioned. There is a menu system to skip to various parts of the video after this introduction. And this introduction is over, so let's go ahead and jump to it. Let's do it! Okay, so first things first, what is glycolysis? Again, we have to understand what in the world glycolysis is before we can actually move on. Glycolysis is a major metabolic pathway. It is much larger and much more used than, well, I guess I could say it's much more used than my previous other videos, which cover glycogenesis and glycogenolysis. Glycolysis is far more talked about and a huge part of our metabolism. It is the first part of our metabolism, and I'll go over briefly the other parts of our metabolism at the very end of this video in what happens after glycolysis. But all you need to know is that it is a major metabolic component, and glucose starts, so once we've digested and absorbed glucose then enters the cell, and that is where glycolysis occurs. And I kind of gave the answer for the next section, but I'm going to go into a little bit more detail there. And glycolysis then acts on that glucose molecule, and we can create energy out of it. So it is a major metabolic component and is absolutely necessary. Where does it occur? Kind of already gave it away in the previous section, but in general terms, glycolysis occurs in all cells, all human cells. It also occurs in other microbes and other mammals, all kinds of other species, but since we're focusing on human physiology, it occurs in all cells. However, not only that, talking about a cell, like if you were talking about different organelles, glycolysis occurs in the cytoplasm, which is that fluidy stuff inside of the cell that's outside of the organelles. So you have different organelles. You might be familiar with biology, microbiology, talking about like the nucleus, talking about mitochondria, ribosomes, stuff like that. Those are all organelles, and the fluid that's between those organelles is the cytoplasm. That is where glycolysis occurs. So you might be wondering, why does this even matter? Why are we even talking about this? Like, glycolysis? Glycolysis who? Who cares? Well, it matters because if you don't have glycolysis, you die. Dead, like straight up dead if you don't have glycolysis. Because glucose gets acted on by glycolysis, and glucose allows us to create energy. Allows that molecule of glucose, and we'll go over that in the next section, will allow us to create energy. So pretty important because we need energy to do this and to talk and to think and to see and to hear and do all these wonderful things. So it is imperative. We have to have glycolysis to live. That's it. That's the reason why we care. Okay, how does glycolysis occur? I'm switching to a blackboard method here. There's going to be another menu, so you can skip through different various sections, of course, of the actual blackboard method, because I know this section is going to be really, really, really long. That being the case, I've stuck another menu in here, so a second menu. So you can thank me later. I'm just kidding. I'm just joshing you. Yeah, alright, let's do it. Okay, so before we get into each individual step, I wanted to have an overview of exactly what we're going to be looking at. And this is the entire glycolysis mechanism, the entire pathway kind of detailed out within the 10 steps that we'll be going through. We're going to truncate it down to 9 because I take step 5 here, or actually step 4 here, is 4 and 5 are truncated into one step when I go through the explanation in just a little bit. But what I do want us to understand before we actually start is that we're starting from a glucose molecule here, and we're going to be going down to a pyruvate molecule here. And as our end result, once we go through this entire process here, we're going to end up with 2 adenosine triphosphates molecules. We're going to end up with 2 NADH, and we're going to end up with 2 pyruvate molecules. And that is what we will get from a glucose molecule. And from there, now that we actually understand what the outcome is, we can go through each step. Now, before we actually go into each step, I do want to clarify that I'm not just going to be going through the mechanistic steps just like every other video is going to be doing that. We're gonna obviously go through each step, but I think what's going to be unique about this is we need to understand, well, we don't need to, but it's interesting, and it's better for us to understand why in the world our body has to go through every single one of these steps just to end up with a pyruvate molecule. Why in the world would our body go from one glucose molecule? Why does it take 10 different steps, according to this chart, 10 different steps to end up at the pyruvate molecule? Well, if we break down each step, which we will understand the reason why behind every single individual step. And I think that's going to be really important to understanding why glycolysis occurs the way that it occurs. Okay, that said, let's go ahead and move on to the first step. Let's go ahead and change glucose to glucose 6-phosphate. See you on the other side. Okay, so we are on our first step of the glycolysis process, and we are starting, our first starting molecule is probably one that most of you are completely familiar with, which is glucose. Now, glucose at this point, Oh, well, I guess I should quick say this is a cell. Okay, so we've got a cell here, kind of a basic structure of a cell. We've got our membrane, which holds everything in. We've got our organelles, this would probably be the nucleus. And then, as it's already displayed here, this is the cytoplasm. And by now you should know, this is just kind of a reiteration of what I've already said, but you should know that this process, glycolysis, occurs in the cytoplasm. So this fluidy texture between the organelles is the cytoplasm, and that's where glycolysis occurs. Now, at this point, however, we are with glucose. So I'm going to illustrate glucose with these terribly drawn orange dots, and we are outside of the cell. So we are not inside of the cell, we have to move inside of the cell. So that's actually exactly what happens. It actually goes across the membrane. So these glucose molecules go across the membrane and enter the cell. So now we have a bunch of glucose molecules on the inside of the cell, and this is of course where glycolysis can then start to occur. However, the reason why I'm telling you this is because this is an integral part to understand why this occurs. Why does glucose have to go into the cell, and then why does it have to be changed into what's called glucose 6-phosphate, as we see here in the first step? Well, the reason is because if we saw glucose just go into the cell across the membrane, it is actually then possible for glucose to then exit back out of the cell. We don't want that to happen because we want glucose to stay in the cell so that we can phosphorylate it and so that we can keep moving on with glycolysis. So to do that, we end up employing the help of a particular enzyme. And that enzyme, well, actually two enzymes, but one enzyme in particular. So if we're talking about muscle cells, then we are talking about an enzyme called hexokinase. And if we're talking about liver cells, then we are talking about an enzyme called glucokinase. And they both do the same thing. So we just take this general cell, and these enzymes then act upon glucose and phosphorylate it, which means that they add a phosphate group to it. Now, anytime I'm going to be talking about ATP, so adenosine triphosphate, which is usually the thing that we use to phosphorylate things. So we're going to be using that energy molecule. You might know it as an energy molecule, and it is an energy molecule. Anytime I mention ATP throughout this video, I am talking about a combination of ATP and magnesium, because magnesium actually needs to be connected to ATP for any of these enzymes to recognize it and then use it for any of these processes. So I'm not going to explicitly say it, but just know that anytime I mention ATP, I'm also mentioning magnesium attached to that ATP. And we are going to see a little bit different circumstances as we go through these steps. Anyways, so hexokinase and glucokinase, depending on where you are in the body, they end up phosphorylating this glucose so that once it goes inside the cell, it is trapped there because its conformation has changed. It no longer looks exactly like a glucose molecule. It is basically a glucose molecule with a phosphate group kind of attached to it, kind of on the side. Actually, on the sixth location of the glucose. So the sixth carbon of the glucose molecule has a phosphate attached to it. And that makes it so that it's impossible for the cell to then go back out of, or not the cell, the molecule, glucose 6-phosphate molecule, to then go to diffuse back out of the cell. So that is the reason why, and that's how it is done. That is the enzymatic reaction. Now, before we continue, I'd like to quick look at some more specifics. So what we're going to do is kind of reiterate exactly what I just went over. Might seem kind of unnecessary at this point, but we're still going to go over kind of the chemical structure and understand what happens. So in this case, we have our glucose molecule, and this is just a normal glucose molecule, and then we end up with a glucose 6-phosphate. So we can see that this is the sixth location, the sixth carbon. It's not explicitly written there. If you know anything about chemistry, you know that these structures don't explicitly say carbon on every single spot that they are located, but just know that this is the 6th location, the 6th carbon of this cyclic molecule, and we have a phosphate group that's been attached to that location. And the way that was made possible was because ATP was taken, again ATP with, I guess I can just this one time write it out, MgATP was used, and a phosphate group, because ATP is an adenosine with 3 phosphates, adenosine triphosphate. So we're taking 3 phosphates using hexokinase, and hexokinase is going to put one of these ATPs, because there are 3 phosphate groups here, and going to place one of them here, and then that's going to leave us with 2 phosphates left. Basic math. ADP, leaving us with ADP, which is adenosine diphosphate, di meaning 2. So this is where we are. This is the molecule that we end up with. Alrighty, so the second step to the glycolysis process is a little hard to illustrate, but I'm going to give it my best shot. Really, the best way to look at it is from the chemical structure, but before we do that, we're going to go ahead and try to understand why this step occurs. Again, like I promised. So the second step is actually a reversible step. There are several reversible steps in glycolysis, but this is probably one of the most important reversible steps, and that's because it is based off of concentration. So this step occurs in the forward reaction if the concentration of, specifically of glucose 6-phosphate, is higher than the concentration of fructose 6-phosphate, which is actually what we are trying to end up with. In that case, then the reaction will be moving this way, trying to make more fructose 6-phosphate. However, it is also possible, and although this is less common, it is more common for the reaction to move the way that I just described it. It is also possible for the reaction to move in the reverse direction, meaning that if there's a higher concentration of fructose 6-phosphate, then the reaction, ooh, that's a bad arrow. That's still a bad arrow. It's a terrible arrow, but let's continue anyways. The reaction will then move in the reverse towards the creation of more glucose 6-phosphate. Now you might be wondering, why in the world would glycolysis then move in the reverse? Well, if you have an overabundance of fructose 6-phosphate that hasn't gone through glycolysis yet because maybe there is less of an energy demand, then glycolysis can in certain steps move in the reverse to kind of back up the system. And that way glucose 6-phosphate can then be used for numerous different things, but specifically in the process of glycogenesis. So the creation of glycogen would be one possible outcome in case you have too much fructose 6-phosphate, which means you have an abundance of energy or energy substrate. So then that way you can then move in the reverse direction and use a different pathway because glycolysis isn't moving at a quick enough pace. However, in most circumstances, in general, we are talking about a increase or a higher amount of glucose 6-phosphate as opposed to fructose 6-phosphate, which means the reaction would go down the glycolysis pathway towards step 3. Now, how is this possible? Well, it is possible due to a particular enzyme called phosphoglucose. Sometimes it's actually called reverse glucophospho. Phosphoglucose isomerase. Now, I think that's important. I just want to quick point this out. The reason why it's called isomerase is because it's going to isomerize glucose 6-phosphate to glucose 6-phosphate, and you'll understand that a little bit more in a second. But really, we're taking both of these molecules, this molecule and this molecule, both have 6 carbons, 12 hydrogens, and 6 oxygens. How is that possible? Well, they are isomers of one another. They have the same chemical makeup, but their structure is different. And that's why it's called an isomerase, because it's going to end up isomerizing the glucose 6-phosphate to a fructose 6-phosphate without changing this component, so without changing what it is made up of. And we will see that right now. Okay, so here it is. When we start out with a glucose 6-phosphate, as you can see, the arrows are moving both ways because, again, this is a reversible reaction, although it tends to be more towards the fructose 6-phosphate pathway. And this is really telling because the structure is the exact same. So as far, well, not the structure itself, the configuration is different, but if you were to count it out, again, not all the carbons are shown here, but if you were to count it out, you would get 6 carbons, 12 hydrogens, and 6 oxygens, and then a phosphate, actually, as well, because we've already added that phosphate from step 1 using hexokinase and glucokinase. So in this case, what ends up happening, I'm not going to illustrate this too much, but phosphoglucose or glucose phosphate isomerase, however you want to say it, ends up breaking one of these bonds and ends up kind of, in a way, kicking out one of the carbons kind of outside of the cyclical nature. So fructose tends to look like this, right? And glucose tends to look like this. So a little more robust. And you can see the difference because this carbon is on the outside as opposed to when it's over here. So it's one location out here. Everything else roughly stays about the same, but the configuration is different. And that is going to set up the next step, as well as allow, just because it is just an isomerization, then that's possible that the reaction can then move in the reverse if it needs to, which it seldom does. And that is that. Let's move on. Alrighty, step 3. We now have a fructose 6-phosphate molecule, and we're still, every step after the first step is in the cytoplasm of the cell, whichever cell you're talking about. So we still have a fructose 6-phosphate, and we are trying to move to a fructose 1,6-biphosphate. So the reason why this step is so important is because it is a rate-limiting step. It is the slowest step of the entire glycolysis pathway and dictates the speed of, well, in a manner of speaking, dictates the speed of the glycolysis pathway. It does that by being affected by the relative amounts of ATP that are in the cell. So if you have high ATP levels, oh man, my arrows are so bad. High ATP, so adenosine triphosphate, so our energy molecule, our most basic energy molecule, if we have a high ATP levels, that is actually going to inhibit our enzyme, which we'll talk about in a second. It's going to inhibit our enzyme from actually going through this process, this step 3 process, and will actually slow down the reaction or slow down the speed of glycolysis. And it does that through a process called allosteric inhibition. Now, if you'd like to know more about that, I have a section dedicated to it in the article, which is linked in the description box. There I explain things a little more succinctly, and if I explained it here, it would take probably a good 3-4 minutes that I don't want to go off track. So just know that if you have high concentrations of ATP, then you end up dealing with allosteric inhibition of the enzyme, and therefore the reaction moves even slower. However, if you have low levels or moderate levels of ATP, then the enzyme functions as normal. That being the case, we can quick look at the actual structure here. So we're looking at fructose, where we have 2 fructose molecules either way, and we have a phosphorylation of the 6th carbon in that fructose molecule, and that's the same, except here we end up having a biphosphate, which we don't have here, and that is because this means 2. So we've phosphorylated 2 different sections of that molecule. So this fructose molecule has been phosphorylated at the 1st carbon and the 6th carbon. Now, in general terms, how does that look? Well, our fructose 6-phosphate, which is illustrated in green here, hopefully you're not colorblind, we have our phosphate, just one phosphate attached at the 6th location. Now, the fructose 1,6 biphosphate has a phosphate attached at both the 1st and 6th carbon. That said, how does that occur? Per molecule, so this is, you know, we're looking at 3 molecules, you know, switching to 3 molecules. Again, this is per molecule. We will be using ATP again, so this is the second step in which we use ATP, just like the first step, and we end up with ADP, so adenosine triphosphate, adenosine diphosphate. So 2 phosphate groups here, 3 phosphate groups here. And the way that works is by using an enzyme called phosphofructokinase. So this enzyme uses ATP, so attaches to an ATP, or well, a magnesium ATP. I'm just going to quick throw that in there, although you should know that from the first step. Magnesium ATP plus a fructose 6-phosphate, and combines and takes a phosphate group off of it and attaches it to that fructose 6-phosphate to create fructose 1,6 biphosphate. Now, let's look at the actual chemical structure of that. So here we are, and we have our fructose molecule, which we know looks like this, right? This remains the same. Fructose here, and we also have a phosphate group. This is a phosphate group on the 6th location. So 6 and 6. Now the difference, of course, is using that ATP, one of these phosphates then the molecule gets changed to ADP as phosphofructokinase acts on this reaction and then puts another phosphate group on the 1 location of this fructose molecule. And that's all you need to know. That is basically it. We now have our fructose 1,6 biphosphate, and we can move on accordingly. Okay, folks, for this step you will probably have to bear with me because I'm breaking this step into 2 parts. Most glycolysis articles, videos, and stuff will name these steps as step 4 and step 5, but I think they're so closely related that they're basically just, there's a step 4 and then a step 4b, so kind of an extension to this step. So my explanation for why this happens is going to be incomplete, but you'll understand in the 4b section of this step when we get to it. So this step, we're going to be converting fructose 1,6 biphosphate, which we created in step 3, to dihydroacetone phosphate and glyceraldehyde 3-phosphate. Now, that said, only one of these molecules is viable to continue through glycolysis. So, and that molecule is glyceraldehyde 3-phosphate, so the purple one here. Why? You will find out in the next section. But in general, just so you know that the breaking of fructose 1,6 biphosphate, which is kind of a different version of our original glucose molecule as it's gone through different iterations, of course. So we'll just, fructose 1,6 biphosphate was, again, that 6 carbon, 12 hydrogen, 6 oxygen molecule, and then we added a phosphate to it, and now we've added 2 phosphates to it. So now it's a C6H12O6 2P molecule. Well, in this case, we're going to break it up into 2 different sections, and these 2 smaller molecules end up playing a role in actually maximizing the glucose molecule. So we're going to be able, even though right now at this point we can only use glyceraldehyde 3-phosphate, we will be able, I might just as well say, we will be able to use dihydroacetone phosphate in the next step. So that's as far as I'll go with that. Now, the actual enzyme that plays a factor in this is called fructose biphosphate aldolase, which reacts right here. So that's all you need to know for this step. Now, of course, again, we're going to go quick look at the actual chemical structure, just so you get kind of a visual of what all this looks like. So here, I apologize for the small image. This is really the best one that I could find that really looked kind of clear. So again, here we've got our fructose molecule, which we by now hopefully are pretty familiar with. Our fructose molecule, which we switched from our glucose molecule in the first step, or the second step, and we've added our 2 phosphates to make a biphosphate system. Now we use aldolase. It's shortened, the fructose biphosphate aldolase is shortened as aldolase in many circumstances, and we've come up with these 2 molecules, which actually, strangely enough, have the identical chemical structure. Not in terms of their actual structure, but more of their makeup. So they both have, and you can count this, of course, they both have 3 carbons, 7 hydrogens, 6 oxygens, and a phosphate group attached to them. So this is found in both of these, although they sound like completely different molecules and their configuration is completely different. They do have the same chemical makeup. So again, that goes back to the concept of the isomer, and that's actually what we're going to use for the step 4b explanation of how we're able to use both molecules, although glyceraldehyde 3-phosphate at this point is the only molecule that can continue down glycolysis. Okay, and as promised, now we can begin to really understand why step 4 occurs. And the main reason is because we started with a glucose molecule and we changed it to a fructose molecule, and then we made, different iterations of that fructose molecule, moving from, of course, fructose 6-phosphate, then fructose 1,6-biphosphate, and then we ended up creating 1 glyceraldehyde 3-phosphate, which, like we said, can go through glycolysis. So its structure, the way it's configured, it is possible for it to go through glycolysis the way that it is, once it's been synthesized from fructose 1,6-biphosphate. However, on the other hand, dihydroacetone phosphate cannot go through glycolysis unless it is switched or changed to glyceraldehyde 3-phosphate configuration. And the reason why that's important is because it is possible to do that using an enzyme called triose phosphate isomerase. And again, here we have the word isomerase, and that's because we know that the structure of dihydroacetone phosphate and glyceraldehyde 3-phosphate, the chemical makeup of those 2 is the same. So we have 3 carbons, 7 hydrogens, 6 oxygens, and a phosphate on each one. And because that's the case, then this enzyme really just rearranges dihydroacetone phosphate to create glyceraldehyde 3-phosphate, which is isomerization. So we are going through that process yet again. However, the reason why this is important is because we're basically branching one molecule. You would have thought that through glycolysis, okay, we use a glucose molecule and it goes through the entire glycolysis system, and then we just get one molecule back out. Well, actually, in actuality, kind of like what we went over in the overview portion at the very beginning of this, of the how-to section, we saw that we get 2 pyruvate molecules and we get 2 ATP and 2 NADH, and we'll go over that a little bit later. But the reason why that's possible is because of this split into 2 smaller molecules. So the glucose molecule is a larger molecule, and then it gets broken up into these 2 smaller molecules. And the reason why that's important is because instead of just getting one product out of that glucose molecule, the body is then able to efficiently separate into two smaller molecules and then get more of a product, get double the product out of that one glucose molecule. We end up with 2 products. So that makes it far more efficient. So this step 4, the reason why that's important is because this splitting into 2 smaller molecules allows for a doubling of the total product when it comes to the end of glycolysis, as we will see. So that's what I kind of wanted to get across. I just wanted to make sure that I kind of explained it well enough. So I broke it up into 2 parts. Step 4b is typically step 5 in most glycolysis lectures, so just be aware of that. Every step after this will be a step behind other lectures, but we are still covering all the same information. Now, to just quick look over the chemical structure, I mean, really, there isn't too much to go over here. If you go back to step 4, just step 4, not 4b, you'll see that this structure and this structure get created out of fructose 1,6-biphosphate, and through this triose phosphate isomerase, we're able to reconfigure this to end up as glyceraldehyde 3-phosphate. Okay, 5th step of the glycolysis process. By now we have converted everything, both of the submolecules that were made up from fructose 1,6-biphosphate into both of them are now glyceraldehyde 3-phosphate, which, as we know, is the submolecule that can then continue down the glycolysis pathway. And from here, we're actually going to be trying to convert it to a 1,3-biphosphoglycerate, which is a different molecule. So it's not the same like fructose 6-phosphate to fructose 1,6-biphosphate, the similar structures with just an additional phosphate. In this case, we're talking about 2 different molecular structures, but there is also an additional phosphate included in this. Now, the reason why this step is important and why it exists is, 1, to set up for the next step, which is really true of most of these steps, but also because we are going to be reducing NAD to NADH for fermentation. Now, I'm not going to go too much more into that. We will touch on fermentation at the end of this video, but fermentation is really through the anaerobic system at the very end of glycolysis. Also, we're going to be using an inorganic phosphate to attach to the biphosphoglycerate. We're not going to be using an ATP like we normally would. Now, NADH is also really important because it's important in fermentation, but also, if you're familiar with the Krebs cycle, NADH also plays a role and is also an integral part of the Krebs cycle, but that's a whole nother subject for another time. NAD is also, in long terms, is named nicotinamide adenine dinucleotide. Definitely a mouthful. Could I say that 10 times fast? No, I couldn't. Okay, so that's why. That was really just a setup, also to create some NADH for fermentation, and also to attach this phosphate, again, really just to set up the whole process. So if we look at these 2 molecules, we can see that not only are we adding a phosphate group, which we will be adding a phosphate group here, It will not be coming from ATP like in previous steps. It'll be coming from it's an inorganic phosphate, which means that it's not attached to an organic molecule. It doesn't have carbons attached to it. So it's just by itself a free phosphate group, which then gets attached to this 1,3-biphosphoglycerate molecule. Now that's all made possible because of this enzyme called glyceraldehyde 3-phosphate dehydrogenase. So really, if we just quick scroll up here, it's basically just taking this name and adding dehydrogenase to it, and there is a particular reason why it's called dehydrogenase, which we will actually go over right now. Let's go look at the actual chemical structure. So when we look at the chemical structure, this is kind of a basic view. The P's are supposed to represent, obviously, phosphate groups. You can see here that we start out with glyceraldehyde 3-phosphate, and then we add a phosphate using again, the glyceraldehyde 3-phosphate dehydrogenase, and then we also reduce NAD+. So when we reduce NAD+, we're actually going to be taking a hydrogen atom and attaching it to this NAD to make NADH. And really this dehydrogenase, if you think of the name dehydrogenase, you're dehydrating, so you're taking away a hydrogen from the molecule, and that's exactly what happens in this case. So you end up losing a hydrogen, or you end up giving up a hydrogen to this NAD to reduce it, which is, in chemical terms, that's what it's called, reducing. So you're going to be adding that hydrogen to the NAD to make NADH, plus an additional proton that gets taken off as well. So this phosphate group gets attached, NAD gets reduced and goes off by itself. Now, this phosphate group gets attached up here, so we have a similar structure from right about here as we do here, but things change once you get up here at the addition of an oxygen and adding a phosphate group as well. Step 6. Moving from 1,3-biphosphoglycerate to 3-phosphoglycerate. So this is kind of the reverse reaction of when we move from fructose 6-phosphate to fructose 1,6-biphosphate. So the actual molecules themselves will look, again, something, I'm not going to draw them all out, but you have 2 phosphates on this one, and you just have 1 phosphate on each one of these. Now, that said, how does that occur? Well, we're going to be using ATP, but we're not going to be using ATP in terms of actually using ATP to add phosphates. We're going to be using ADP to move a phosphate from these molecules here, and then we're going to just have 1 phosphate left. So basically this step, and the reason why I'm telling you this is that the reason why this step is important is because it is the first step where we actually see an increase in energy. So we end up creating energy, synthesizing energy due to this step. Having an overabundance of phosphate groups on this molecule allows us to then use ADP. So we end up with ADP here, again 2 phosphates, and ATP, adenosine triphosphate, with 3 phosphates. So this loses a phosphate so that it can become 3-phosphoglycerate. Now the enzyme that actually acts on this is called phosphoglycerate kinase, and it acts on the reaction to take away one of those phosphates and then deposit it onto the ADP and make ATP. Now that's really basically it. You're just creating energy through this step as well as furthering the reaction, as with most others. Now to quick look up here, you can see here we've got our 1,3-biphosphoglycerate, we've got our two phosphate groups. And here we're down, we've given up a phosphate, so we only have one phosphate left, and we still have this oxygen up here. So that's purely it, just giving up a phosphate to create energy, 1 ATP per molecule. Step 7, moving from 3-phosphoglycerate to 2-phosphoglycerate. Hopefully you can just look at the two names and be able to tell that really nothing changes here. The chemical makeup of these two is identical. We are going to be moving a phosphate group from the third carbon to the second carbon. That is it. So in terms of actually looking at it from a general perspective, really there's nothing to be said. There's really nothing to be done here. Just knowing that we're going to be moving from this 3-phosphoglycerate, moving a phosphate group, and we'll see that once we look at the chemical structure, but there's nothing major that occurs. And the reason for this step is really just to set up for the final steps. The final two steps, this step, it's important of course because it is part of glycolysis and it's still an integral part of glycolysis, but does it have some flashy movement or anything like that? No, it's probably the most mundane step of them all. So the actual enzyme that plays a factor in this is called phosphoglycerate mutase. So without further ado, we can just go ahead and look up at the chemical structure, and here it's really simple. I mean, everything is the exact same. You know, you look at the main body of the molecule and everything is the same. The only change is this movement. This phosphate moves to this carbon over here, and that's it. That's the bottom line. Really, this step, all it is just a setup step for the following two steps. Okay, so the second to last step of the glycolysis pathway is moving from a 2-phosphoglycerate to a Phosphoenolpyruvate. In this step, the reason why this step is important is because we are going through yet again a dehydration process. We are actually going to be taking away hydrogens and an OH group to create water. Not only that, we are also going to be destabilizing the molecule, which might seem like a bad idea because then you've got an unstable molecule, but actually in reality what that does is it creates a more energetic environment so that we can garner more energy from that molecule. So it's a little difficult to actually show that in the general picture, the big picture, but what we end up doing is getting an H2O out from this molecule so that we can end up with this molecule. Now the way we're able to do that is by using an enzyme called enolase, or enolase, however you want to pronounce it. So enolase acts on the 2-phosphoglycerate to then extract the hydrogens and the oxygen to create phosphoenolpyruvate, which is the second to last molecule that we will see in glycolysis. However, for enolase to actually work, we need to actually use magnesium, just like we talked about when we talk about ATP, for example, you know, magnesium ATP. Well, in this case, magnesium has to attach to enolase to actually make enolase, the actual activity of enolase go through so it can actually go through this process. Okay, so actually looking at the chemical characteristics, here we can see that not a whole lot changes. But we do end up releasing this OH group, and then we end up releasing this hydrogen so that we come up with H2O. Now you also notice that we have a double bond here because we're changing the bond structure so that we can destabilize this molecule, but that actually makes it more energetic, or be able to release more energy later on. So that's the purpose and how all this occurs, through the enzyme enolase, which is activated by magnesium, so free magnesium. And in this video, this is the final step, step 9. Again, just to reiterate, most other videos and most other explanations of glycolysis do not combine step 4 and 4b. They separate them, so there's 10 steps in those. However, just because I felt like it was better, this would be a 9-step process. We still go over the same steps. Anyways, moving over from phosphoenolpyruvate to the final molecule, which is pyruvate. This step occurs because we have a second chance to create energy, and you can probably guess how we do that. We've already done it once before, and we've used it to actually we've used energy this same way, in kind of the reverse way, but we end up using ADP to create ATP, and we're able to do that because there is a phosphate attached to these molecules from the phosphoenolpyruvate. And that's basically it. You know, we're just finishing up glycolysis, we're finishing up the entire pathway, as well as giving ourselves one more chance to create just a little bit of energy out of the energized phosphoenolpyruvate from the previous step. Now, as far as the actual enzyme is concerned, we're talking about an enzyme called pyruvate kinase, which acts on here. Acts on the reaction, and for this pyruvate kinase to actually be functional, we need to attach a potassium and a magnesium to it. So you would need potassium and magnesium for pyruvate kinase to function, so those two would need to attach. So now moving on to the actual chemical structure. Again, I kind of apologize for the small picture, but for some reason there just aren't very many good illustrations. But it seems it's pretty simple. So we end up removing this phosphate, which then goes to create ATP. So we move it to ADP, you know, goes across, creates ATP. So we lose this phosphate, and not only that, we end up moving these double bonds. We pick up a hydrogen here, and we create a double bond, creating a ketone group on this carbon here, which creates pyruvate in its final form. And that is the end of glycolysis. That is the very end. And of course, with pyruvate, then we go through other processes, other metabolic processes, which we will briefly cover a little bit of what we can do in the next section, but for detailed analysis, you will just have to wait until another OmniFocus Fitness video emerges. Okay, all right, we're done. Moving on. Okay, now that you've just maybe sat through a massive amount of information, you're probably just boiling over. So now we know that we've got 2 pyruvate molecules, we have 2 ATP, we've created 2 ATP, so energy, and we also have 2 NADH molecules. But what happens after glycolysis? I did mention that glycolysis is the first step, a major component of metabolism and energy production, etc., etc. But there are other steps. So what are those other steps? What happens after glycolysis has gone through all of its steps, all of its 9 to 10 steps? Well, in that case, it depends on the physiological state of the cell. So if we have oxygen, so if we have sufficient oxygen, glycolysis then goes through the TCA cycle, and otherwise known as the Krebs cycle. So if there's enough oxygen to go through aerobic metabolism, that's what that's called, that's the overarching term, aerobic metabolism, then it goes through the Krebs cycle, otherwise known as the TCA cycle, and then goes through the electron transport chain. However, if we do not have sufficient oxygen, then in that case, then we end up with lactic acid fermentation. So in that case, we end up using NADH, the two NADH that we created along with other NADH, and we end up kind of recycling that through, and we end up making just a little bit more ATP, but of course that's not a sustainable, and it's not a sustainable system for actually creating energy, because it is with a lack of oxygen, which means it is, the overarching term for that is anaerobic metabolism. So the lactic acid fermentation is using NADH is used when oxygen is not present, or not enough is present. So those are the two possibilities, those are the two major possibilities when it comes to human physiology, human metabolism, and that answers that. Of course, for more details, you will have to stay tuned for further videos and articles. And that being said, I've put a lot of work into this article, I've put a lot of work into this video. Please feel free to check out the article below. I have like a ton of scientific links. Everything is sourced out, everything is completely legit, like legit, okay? So please, it would mean a lot to me if you ended up subscribing, because I put a ton of effort into these videos and articles. So if you could subscribe, that would certainly motivate me, and it just makes me feel good to see that subscriber number increase. And without further ado, that's it. That's all I've got to say. I know that's it. It's a ton of information, but hopefully you found it informational, and I will see you in the next video.